Hi, everyone. It's good to see you here and see many of you again. Seen many of you over the past number of years. It's also great to hear from Diane about the OTB and the overview of that program. And I will say that we are more recent users of that activity. And so we've been really enjoying our collaboration with that activity. So, but I'm here to talk about the Ontario Health Study. This is a joint presentation by myself. I'm the PI of the activity. With Kelly McDonald, the Senior Program Manager. So I'm going to start off that presentation largely about our vision, and I'll turn it over to Kelly, and then I'll give you some case examples of how you can use a population cohort like the Ontario Health Study to support research. So this is just a quick snapshot of what we anticipate in terms of public health and population health. So we anticipate these numbers and frequencies with respect to cancers and chronic diseases over time. And so a cohort like a population study like the Ontario Health Study, it is a population cohort. It's distinct from, say, a clinical cohort. And we're largely trying to capture as much information as we can about people in the population as they develop diseases or before they develop diseases or once they've developed diseases. So these are the kinds of questions that we as population cohorts, and we're one of a number of cohorts internationally, where Ontario and Canada were if you like, the population cohorts representing Canadian studies in this space. So we're primarily interested in addressing questions like, what are the root causes of health and disease? What can we do to improve our health? Address questions around health equity in the population. What can we do to build healthier communities? Identify cancers. Can we identify cancers early? We heard a great overview and vision from Laszlo and Lincoln in this space as well. And what can we do using population cohorts to build learning health systems? And so a population cohort that we are building, and we think some of our value is our ability to think both prospectively and retrospectively, because we're a longitudinal cohort. We capture information over time. And the Ontario Health Study, I think, started in 2007. I should know that, but we did start in 2007 in terms of our pilot activities. And we've been collecting information from Ontarians since. So
it's a large population cohort. It's Ontario's largest population cohort.
And because we capture information over time,
we capture information from some people
as they came in
healthy from a particular age range, and Kelly's going to
get into some of the specifics about that. Some of those people came in healthy and then have since developed a disease. And because of our activities in terms of capturing information and how we capture information, we can identify who are those people who developed a disease, which disease, what kinds of treatments did they get, where did they live, what were the potential environmental exposures associated with that? So these are the types of questions that one can ask as well. And we can also ask questions about who hasn't developed a disease. I'm particularly interested in that space. We have research projects around who are our healthy Ontarians, who are our 80-year-olds, who are our 90-year-olds, who have, say, the blood or health profile of a 40-year-old. So we can ask those types of questions too. So this is where I'm going to turn it over to Kelly, and she's going to give us an overview of the OHS and, to some extent, CanPath. So.
Thanks, Philip. So Philip mentioned the workshop and the thought process around developing the Ontario Health Study started back in 2007. Our first participant was recruited on March 26, 2009, right before the end of fiscal year. That pilot phase lasted just over a year. In 2010, we ended the pilot phase. We took a look at what we were doing, how we were doing it, and we revamped a little. So we broadened the eligibility criteria so that any adult resident of Ontario over the age of 18 was able to participate. We moved data collection online rather than asking people to go to specific assessment centers to complete that questionnaire or series of measurements. And we ended our baseline recruitment as well as our baseline blood sample collection in March of 2017. As Philip said, ongoing and longitudinal data collection is the strength of the Ontario Health Study and CanPath, which I'll get to in a second. So we've administered our first follow-up questionnaire between 2016 and 2018. We conducted an occupational history questionnaire in 2019, and then we started our work to support COVID-related activity in 2020. So I mentioned that we ended our baseline recruitment and blood sample collection in March of 2017. The OHS collected a blood sample from over 40,000 participants during that baseline phase. As part of our blood collection workflow, we did a measurement of the complete blood count as well as HbA1c on the majority of those participants. And then in support of some of our COVID-related research, we also collected almost 10,000 dried blood spots from participants from up to 3 dried blood spots from OHS participants over the course of the pandemic. So for over 3,000 participants, that represented a first blood sample to the OHS, increasing the total number of participants who'd provided a first sample to over 43,000. But it also represented a second blood sample collection for over 6,000 OHS participants. The OHS has established agreements with Ontario Health as well as the Institute for Clinical Evaluative Sciences to link the OHS data to the administrative data at those institutions. So that allows researchers to access vital statistics, claims, cancer screening programs, and of course the Ontario Cancer Registry. As part of that, we're also able to actually then select biospecimens for analysis based on whether the participant has been diagnosed with an incident or prevalent case of cancer, or whether they are confirmed as being cancer-free. So this slide represents the incident and prevalent cases of several chronic diseases following a linkage with ICES in about March of 2020, quite timely, given everything that was happening in the world at that point. And so the blue bars represent the prevalent cases and the orange represents the incident cases. So OHS data has been included in about 60 different data sets that have been accessed by approved researchers. This is a list of some of the findings that data analysis has supported. About a third of those applications do support cancer-related research. In addition to finding our access policy online, as well as our access application, you'll also find a lay summary of every approved access request that we have fulfilled for researchers. So some of those approved access requests came about because the Ontario Health Study is part of CanPath. They're the Canadian Partnership for Tomorrow's Health. CanPath is jointly hosted here at OICR and at the University of Toronto, and Philip is its National Scientific Director. So CanPath includes data from over 330,000 participants across 10 provinces, with the OHS contributing about half of that data. And CanPath also has a repository of about 150,000 biospecimens, or biospecimens from 150,000 participants. So this dramatically increases the sample size. And really gives researchers the opportunity to study rarer types of cancer, as well as to investigate a broader range of exposures. The CanPath cohorts have committed to harmonizing their data and biospecimen collection as much as possible. So the biospecimens were all collected and processed using the same protocols, and the core data that's been collected through our common questionnaires has also been harmonized. And over 1,600 variables on disease outcomes across multiple time points are available through CanPath. One of the linkage opportunities that is available both at the OHS and CanPath is through CANEW, the Canadian Urban Environmental Health Research Consortium. CANEW is an enormous database of environmental exposures. So water quality, air pollution, noise exposure, light at night, green space, all of that kind of thing. And participant data has been linked to those environmental exposures at the postal code level. And a couple of the cohorts, including the Ontario Health Study, have completed a historical linkage to construct residential histories so that longer term environmental exposures can be included in analysis. At this point, I will turn it back over to Philip.
Great.
Thanks, Kelly. So as I mentioned before, Canada is CanPath, sorry, and the Ontario Health Study are Canada's largest population cohorts to date. And because we are the largest population cohorts, we have been directly implicated and involved. And in fact, at the OICR, we actually host some of the work associated with the International 100,000 Cohort Consortium, which is a consortium of all population cohorts. With some of the folks include here, 23andMe, UK Biobank, the All of Us program, which are recruiting 100,000 participants or more. And so with our friends upstairs, Melanie Courteau and Genome Informatics, we're actually hosting here at OICR the data portal for that activity, which is this is going to be a fantastic resource. If you're a scientist who's interested in using data for multiple international activities, you want to find all the type 2 diabetes participants in these population cohorts from across the world, you can go to this portal and do a search here. So just wanted to highlight that as a particularly important activity that's ongoing as well. The OHS, because of its size, was actually the first cohort that was invited to this activity, but we decided to bring in all of CanPath to this activity as well. And you can kind of see that up here. So just to emphasize now a number of activities that you can do with things like the OHS and CanPath, given our resources, our consents. Everything starts with the consents of our participants. And those consents allow us and enable us to do those types of linkages that Kelly just mentioned a moment ago, where we can link to administrative health records, for example, and identify in a prospective way, as well as a retrospective way, who's developing what. And in this case, what we're calling the Canadian Cancer Study is an activity that we're starting here out of the OHS and the OICR. That's already beyond starting. We've already started these analyses, and we're hoping to have a submission to a magazine or journal by the end of this calendar year, where we're trying to identify both those incident and prevalent cancer cases across both OHS and CanPath. So those prevalent conditions would be individuals who came into the cohort already diagnosed with a cancer. An incident case would be an individual who came into the cohort and then subsequently was diagnosed with a cancer. And again, we can only do those types of activities through our consents that allow us to link to administrative health records, all of our participants to become an OHS, almost all of our participants to become an OHS and CanPath participant, had to give us health insurance numbers, other identifying information like date of birth, names, and so on. And we can use that information to link to these various registries across Canada. So right now, active work right now, including partnerships with ICES, Cancer Care Ontario. And right now we've got data coming in from these 4 regions as well. We're mapping the cancer cases in the CanPath cohort. These are the cancer cases that are accumulated that are both, I think these are the total cancer cases across these 4 provinces at present. So I'm not going to go into all of these numbers. So this is coming directly from the cancer registries themselves from each of these provinces. Now, while we're doing that in each of those provinces, we're very excited that here in U of T, we are now signing an agreement with the Canadian Institute of Health Informatics, where we'll be able to collect, and we're at signature stage now. So where we'll be able to host all of the provincial data that CIHI, the Canadian Institute for Health Informatics, collects about CanPath participants. That's a big deal. The reason that's a big deal is because that's going to be a first in Canada where you don't have to go to 10 different provinces to capture all of this information, which we are doing. We're doing that legwork for you. But now with CIHI, we can have a one-stop shop, if you like, for national data that CIHI has already collected for us, and we'll host that at CanPath. So you can get access to that information. Now, I'm emphasizing that linkage activities because those linkage activities really does help us with scientific activities like early detection of cancer. Because what we've collected in these population cohorts in CanPath and in the OHS are biologics of participants, as we just mentioned, before somebody was diagnosed with a disease or a cancer. So we have the opportunity in a, if you like, safe space to analyze our biosamples to see if
There's a signature in those biosamples and other data as well. It could be other data that you don't capture from the ICD code.
For example, we get smoking history, alcohol history. Where do you live? Do you live
Under a dry cleaner. I don't know.
Dry cleaner. But those types of information is the kind of information we capture coupled with some of the things we can interrogate from blood samples. So I have a very talented student who likes to put these gradual slides into these transition slides in, who's been developing tools largely in collaboration with our friends at PMH and UHN to develop cell-free methylation signatures to see if we can see a signature in these plasma samples of the OHS of an early cancer signature.
Right. We like DNA methylation because it gives us a snapshot. It's very tissue specific DNA methylation. So you get a sense, not just that there might be a signature of a cancer, but potentially where that tissue came from as well.
Where that cancer is. We've been developing these tools. I'm going to fly through this really quickly because we've developed methylation marks or profiling tools, bioinformatic tools, using, of course, lots of the methylation data that exists in the Cancer Genome Atlas. And as I said before, the Ontario Health Study allows us to identify, say, prevalent and incident cases. So these are the incident cases for breast, prostate, pancreatic, and hematological signatures that we've started to explore as they're accumulating in the Ontario Health Study. So you can see it's a fairly linear rate. And this is why, again, population cohorts, you have to build them, you have to invest in them now, because their real power and value increases with time, because unfortunately people start developing these cancers over time. So as you can see, these numbers are increasing over time. And so what Nick did is identified the incident cases of breast, pancreatic, and prostate. And what we also have information of is, when did somebody give us a blood sample? And then when were they diagnosed with the cancer? Because then we can go back and see how early can we see a signature? How early can we go? We can only go as early as the cohort's been around. So we've been around, as Kelly mentioned, for a period of time. And, some of the cancer registries data is lagged. You can only, it takes time to build cancer registries. They're really good. There are people who develop the algorithms for that. And it's about a 2-year lag, in fact. So this is just data for 2017. We've done another poll for 2019 cancer registry data. So in fact, we've doubled our cohort size. And so you can see on that bottom figure, this is when a blood sample came in, and then this is when somebody was diagnosed with the cancer, right? So sometimes somebody gave us a blood sample and we're diagnosed like a month or two later. Other cases take maybe a number of years. And we also have information about mammography as well. We have information about stage, grade. We have information from those cancer registries about pathology. So we've been using those blood samples, as I said, from plasma samples in particular, cell-free, to see what we can do. And so this is just a really quick picture here, principal component analysis, just to show you that from our plasma samples, you get really decent separation of cell-free DNA methylation signatures in, say, breast, prostate, and pancreatic. This is just our first poll set compared to controls. And again, that's where the Ontario Health Study comes in handy, because we have a lot of data for controls. We can do age match, we can do type 2 diabetes match, we can do sex, obviously sex and gender matches as well. And yeah, so you can actually see that. So, and of course, breast and prostate, we have the most data for, because those are the highest incidence rates. Pancreatic, very few. There's only a small number, but that's where CanPath becomes important, because to really support and power a study for a rare disease, you need to be looking nationally as well. Nick's been able to show with his work that he can actually discriminate luminal A and luminal B breast cancers in the data. And one of the things that I wanted to highlight really quickly, our accuracies under the curve, our general overall accuracies, I'm just going to fly through a couple of these, are around 83% right now. We think that's really good. Our false positives are pretty low. Our false negatives are still not at a place where I think any of these tools are clinically irrelevant. And in fact, I was just at UK Biobank, where they're supporting, not UK Biobank, at Oxford, where they're supporting these massive clinical trials for the GRAIL activity. And those, Lincoln and I were talking about this yesterday. Those numbers are still underwhelming. Those activities, I think, in the liquid biopsy space, getting out into the commercial space, I think should be coupled with mammography. They shouldn't be replaced with mammography or traditional diagnoses. With that said, our signatures can see a signature of the breast cancer even when somebody did have a mammogram. So within months, right? So there was a signature that we were able to capture even in our participants who had just been diagnosed with weren't diagnosed, with breast cancer, the mammogram didn't detect it or the clinician didn't detect it, we did see something in our data as well. And of course, these tools work really well when the cancers are more advanced. And so you can see that the areas under the curve, naturally, when the cancers are more advanced or metastatic, there's a lot of shedding of tumor material and blood. So naturally, we're seeing this. I just should emphasize, of course, that all of this was done here at the Genomics Institute with our genomics colleagues, with Bernard's team and Trevor's team. And and we've got some, I think now, significant expertise in running these types of liquid biopsy assays. The last thing I'm going to highlight here is just to emphasize, I just talked a lot about plasma, but I just graduated a student, Alyssa Bader, this year, and during the course of her PhD work, she was also able to demonstrate that blood samples collected in the Ontario Health Study can support single cell assays. That was also work done here at Genomics. We've been doing a TAC-seq, we've done whole genome seq, and this is just a snapshot. I'm not going to get into the science of this project, but we were pretty excited about this because this is one of the first population cohort to do this type of assay at scale. So I think am I turning this back over to you? The next slide is this one. So okay, that's fine. We'll just end here. So again, that's just to give you an idea of what you can do with the biologics and the kind of science that we can do. As Kelly mentioned, we've had a number of projects come in to use the OHS. The OHS has also been used in a number of those CanPath studies as well, and well over 300 projects have been supported by CanPath as well. So, we do support scientists in terms of trying to get access to samples, their grant writing, their leveraging of our cohort to get the resources they need. This is just a snapshot of how we've been able to support mostly national research. We do support international research as well. And okay, this is where I'm turning back to. Okay,
So this is how you actually go about accessing OHS data and biospecimens. If you actually, our access officer is at our booth upstairs and she could give you a handout of exactly what's available, number of samples, serum, plasma, red blood cells, Buffy code, DNA, genetic data, the single cell data that Philip was talking about. So all of that is written down and in a handy little handout. All of this information is also available online and there's a short video that walks you through it. The process is pretty straightforward. It's very similar to the Ontario Tumor Bank. We strongly encourage people to reach out, start a conversation, make sure we have the data that you are looking for, depending on what data or samples you want and whether it includes linkage. There's a bit of a different process depending on who we're linking with, et cetera. There's an access application that gets completed. We need evidence of RAB approval, short CV. If your research project has been peer reviewed for a funding application, for example, we'd like to see evidence of that as well if it exists. It's adjudicated by some independent reviewers. There's an access agreement, and then we start the conversation around prepping the data set and transferring it to you once those agreements are in place and the fees have been paid. So as Philip mentioned, it takes a village. So this is a whole host of organizations in addition to OICR who are very supportive of the CanPath cohort in particular, but also the Ontario Health Study. This is the OHS team. Currently, we are a group of 15 very hardworking, dedicated individuals. About 4 of the people on that screen have worked with the OHS for over 10 years. Speaking to the commitment of the staff to the project. And this is Philip's lab. He brought forward several of the projects that OHS and CanPath data have supported through their work.